Awesome. So as mentioned, my name's Kevin Cook. I'm research faculty at the Developing Brain Institute at Children's National Hospital down in DC, where we work in fetal MRI as well as neonatal and preterm MRI. But today we're going to be starting very early, looking at functional MRI still in utero during the fetal period. Because I am First up today, I am just going to spend a second justifying why we're talking about this today, why study early life, why is it important, rather than just starting in childhood, in adulthood. And in addition to just growing our knowledge, wanting to know more, there is this really central theory of the developmental origin of health and disease. Formerly, you might have heard fetal origins of adult disease or prenatal origins of adult disease. But the idea is that developmental foundations are really laid at the earliest stages of life, really within the first year. So from conception until about the first birthday, most of our developmental foundations are laid. So our fundamental brain development is really supercharged in the first year of life. And there are lots of things that impact that development, right? So genes and environment are two huge factors that play a role in how that foundation is laid. And when I'm talking environment, I'm talking both our broader environment, so our socioeconomic environment, our neighborhood environment, but also the maternal fetal environment, right? And importantly, a lot of prenatal conditions are associated with later on issues with cardiovascular disease, with diabetes, with obesity. So anytime anyone talks about this research, they're going to mention the Dutch famine studies, which is one of the most well-characterized set of studies looking at how conditions during the prenatal and early neonatal period have knock-on effects throughout the lifespan. So this was tail end of World War II in the Netherlands and German-occupied Netherlands, where there was a famine that importantly was time-locked. So it was a very discrete period, one winter. It was also geographically locked to this one very specific region, which allows for a naturalized study where we can say, okay, we can compare this to similar groups, both in time, so the very next year where there wasn't a famine, or geographically similar regions during that same year. And in addition to all of the issues occurring during that period, these kids went on into adolescence, into adulthood with higher rates of cardiovascular disease, diabetes, obesity, a lot of largely metabolic focused disorders at the time. Importantly, this research has been replicated across other famines as well. So it's not just the Dutch famine, but it's one that is probably the most well characterized. But more recently, there have been tons of studies looking at other types of exposures and other types of risk. In particular, neurodevelopmental psychiatric disorders seem to have a lot of their origins in the prenatal or early neonatal period. So this figure, really nice example, little old, needs to be updated a little bit, but I think it's a good illustration on this idea that a lot of these disorders are ones that are heavily centered in either the early growth and development of brain structure, brain function of synapses, as well as atypical pruning in early life, so excessive pruning or insufficient pruning. And when we think about this, we can see what a big change there is in brain development throughout the fetal period. So this is a set of structural images from about 23 weeks gestation up through 35 weeks, so not even term age yet. And we can see how much the brain is growing and developing during this period. So you see all the way over 23 weeks, it is really a soft inflated balloon, right? It's relatively small compared to that 35-weeker, about a quarter of the size. You don't have much cortical complexity, but you see very quickly, we see the forming of those sulci, of those gyri, of this developing complexity of especially the cortical gray. And really nice paper from Nature a few years ago, which I believe both Chris and Damien contributed to, looking at changes in the brain throughout the lifespan. Of course, today I'm going to highlight just that first little bit, that first about year of life. And we see that this is cortical gray matter. On the left, we have the normative trajectories. On the right, we have the actual growth, so the rate of growth. And we see that really within that first year of life is when so much of that development is occurring in terms of getting that initial volume up, but also the rate of change is so pronounced in that first year of life and then really slows down, making it a really important period for us to understand those later issues, because that is when, as I said, that foundation is being laid that is going to scaffold all future development. So from here, I'm going to talk a little bit about technique of acquiring these images, a little bit about how we process the data, and some very fundamental things that it's important to know about this period. So a very brief history lesson. So the very first fetal MRI was actually in 1983. So very early. I'm always surprised when I see how early that is. But at that period, the focus was really on, is MRI safe? When we put a pregnant person in an MRI, is it safe for mom? Is it safe for fetus? The really first functional MRI looking at brain activity in the fetus was in 1999. Really simple study, right? 1999. It was a simple tone on, tone off task, looking at auditory sensation. Simple, but the first time it was done. But then really the focus shifted in about 2012 to the first resting state study. So this is Daniel Prayer, one of the big names in this early development of fetal MRI. And again, study from 2012, so methods have updated significantly from then. But here we're looking at bilateral connectivity of frontal regions in development across weeks, so from 32 weeks up to 36 weeks. In terms of our institute, we've been collecting fetal MRIs since about 2011. Our first usable ones are not until 2012 because it takes a little while to figure out exactly how to optimize the scans and the parameters, especially back in 2011. We have historically collected 1.5T. We, as most labs have, transitioned over to 3T. We're a little bit slower because there's always a concern about making sure everything is safe for this period that is so sensitive. And we scan mostly in the second through the third trimesters. A couple reasons for that, and I'll talk about that in a little bit. And we've investigated typical development, but also a bunch of adverse exposures of congenital heart disease, CHD, maternal stress, neighborhood and socioeconomic disadvantage, all of which I'll be talking about in a little bit. So here's an example of a fetal functional MRI collection. So this is at about 30 weeks. And you'll notice really right off the bat, the two big technical concerns when we're collecting this data. First is maternal tissue, right? So we have tons of maternal tissue to deal with when we're processing the data. The second is obviously motion, right? So as much as we ask these fetuses to try to stay calm and not move too much, they're not great at listening quite yet. We don't really have the ability to pack their heads. And so we have a lot of unrestrained movement. So this is one scan, one kiddo taken every 30 seconds. So it's only about a minute of scanning. And we'll see in that period here, right, we have a kid who's looking relatively straightforward. We do about a 30-degree turn to the left, and then we do about a 45 to the right, and then pitch up a little bit. This is pretty standard for what we see for our kids. Some are better, some are worse, but we have tons of motion, which is obviously a concern. So we have some ways that we try to process this data that is a little bit different than we do with ex utero, with kids, with adolescents, with adults. So the first step is trying to just identify what brain tissue is. So we have a couple in-house algorithms that do this. And to isolate the brain, mask it. So the first step is we just find where the brain is, right? We try to find a mass that looks like the brain. This is all based on a 3D UNet framework right now. It used to be done by hand, but we identify center of mass of the brain and draw a cylinder around it. So just try to get as much tissue as we can, but try to isolate the brain as much as possible. At which point we then extract it. So you'll see here on the left, it's extracted, and then we reorient it. So you see we maintain that sort of square bounding here, but it's been rotated and translated over to their anatomical image, which has been hand reoriented. We then have another 3D UNet-based algorithm that is going to mask the brain. based on that reoriented image. So we see it over here, and we have a rough probability map. So we have that definitely brain in the center, and then we have that edge around it. And this allows us to create a mask that we can use for subsequent processing. So at which point then we work within bioimaging suite. So that's from up at Yale, Dustin Shinost and the rest of the folks up there have a fetal motion correction algorithm. So that utilizes both that EPI that I corrected before and the mask that we create to frame by frame reorient the image. We then conduct motion censoring, which you'll notice right off the bat is pretty liberal compared to where most ex utero MRIs are. So these are not the parameters, for example, that I would use with our neonates. But as you saw, we have a lot of motion, so we need to be a little bit more generous. So we use 3D outcount to identify intensity outliers. So those volumes that have a lot of those outliers. We set a translational movement over 1 millimeter and a rotational at 1.5 degrees. But then once that's been fully corrected, we largely are going to mirror ex utero pipelines. It's just getting the data to that point. So just as a brief overview, we're going to do some anatomical registration. We're going to do some intensity scaling, some smoothing. We're going to use a 4.5 millimeter kernel, do some bandpass filtering. Going to censor out all those frames that I mentioned where we get the high motion, both the frame, both that volume, the prior and the subsequent, and do some nuisance regression based on a tissue map that we developed for each individual participant. And then once we get that data, we can do most of the same analyses that we're going to do with our ex utero kids, our neonates, our children or adults. And I'm going to highlight a few studies just to also highlight some technical considerations when we think, some theoretical considerations of working with data across this period. So this is some work from Mariah Thomason. We'll be talking about her on a couple of these slides, another big luminary in developing this field. But we're able to do seed-based connectivity work. And what we see is a pretty structured, organized development to connectivity across the fetal period. So most regions are going to develop local connectivity. It's much stronger early on. And once we hit about 30 or so weeks, we start developing more and more of these longer range connections. The brain is also largely going to develop from a medial to lateral pattern, posterior to anterior. And you'll notice that we're starting to form some very early proto-functional networks. So I'll draw your attention to G, that medial PFC. Very early on is going to be mostly limited to this local connectivity within the medial PFC. Once we get close to term age, you're starting to see we're adding in that PCC. We're also adding a little bit more of those lateral components. We start seeing a very, very early form of a default mode network starting to develop, even prior to birth. To talk a little bit more about this network connection. So we can also do broader network level research. So here on the right, we have adults about 30 years of age. On the left, we have those fetal connectivity. This group is going to range from 20 to 40 weeks. So it's a little bit of a mixed bag in terms of age. But what we see here is that a lot of these early networks are mirroring what we see later on. So down here at the bottom, so we have motor network starting to show, again, not as robust as we're going to see in adulthood, but a clear pattern starting to emerge. See the same thing with default mode, again, not as robust as adults, but starting to show up in visual as well, really starting to develop. A little bit weaker in terms of other networks at this very early age. Subsequent work has found that, obviously as you get closer to term age, more mature, more adult-like networks are starting to emerge. But it is really driven by that medial to lateral, anterior to posterior, sort of proximal to distal development of connections very early on. And again, we can do the same thing with graph approaches as well. So we see here work from our group looking at changes in graph properties across the fetal period. So here we have clustering modularity, small world local efficiency, basically showing that there is this developing pattern of segregation and integration that changes across gestation. So it's not a flat trajectory over time. These connections are growing and developing. And then I'm going to talk just a couple quick applications. I'm going to try to be mindful and not immediately knock our schedule completely off. So we know a bit about typical development, but these are also, as I mentioned, very important tools to understand atypical development, types of stress. And here we have another study from our group looking at maternal stress and how that impacts functional development, functional connectivity across the preterm period. So here we have one example using the STAY, looking at trait and state anxiety. And we find even prior to birth, mom's level of stress, both from a trait perspective and a state perspective, is impacting development, is changing connectivity. So here in the green, we see stronger connectivity. So those modules that are more connected, red is decreased connectivity compared to as a function of increasing anxiety, whether it's state or trait. Similarly, as I said, we work a lot with congenital heart disease, and we see that functional networks during the fetal period are heavily disrupted by the presence of CHD. So on the left, we have representation of a subnetwork from a network-based statistics model that is associated with congenital heart disease. Importantly, all these connections that we see here are decreased connectivity. So we're seeing less connectivity in those kids with CHD or those fetuses with CHD, not even kids yet, those fetuses with CHD compared to healthy controls. And what's really cool is that on the right, we have those same connections, but looking at its association with preoperative O2 saturation. And basically, I think it's 75% of the connections that we see in the subnetwork are also associated with decreased O2 saturation preoperatively in these kids, drawing this direct connection between the actual phenomenology of what's going on in congenital heart disease. We think it's disrupting oxygen transmission to the brain, and we see a direct link between that oxygen saturation and these developing connections in the brain. And then finally, we've also started to do work in neighborhood and socioeconomic disadvantage. Happy to talk about this a little bit more, but I just want to make sure that we're keeping time. But we see that the neighborhoods that families are in prior to birth is already associated with changes in graph properties in these functional topologies, even prior to birth. And again, just here dependent on what communities mom is a part of during pregnancy. And so across these studies, we see that functional connectivity follows a pretty consistent developmental trajectory, that deviations can be observed in states of stress and disease. And importantly, most of the groups that I know who are doing fetal work are continuing to follow these kids long term because it's nice to know what's going on during the fetal period, but what does that look like behaviorally later? And more important, once these kids are old enough to come back, to start doing some more functional MRIs, whether it's resting state or task-based. Can we see precursors of disruptions later on during the fetal period? So our group is just at a point now where we're getting some old enough kiddos to come back and start doing some resting state MRIs now that they are in sort of middle to late childhood, which is really exciting. And I hope soon to be presenting some of that work as well. And then I just want to call out the entire team at DBI. So we're a very large team. Doing fetal MRI is a complex task that requires everyone from MR physicists to developmental psychologists to make sure that we are all accounting for everything we need to. And so this is our lovely team here. And thank you so much for your time. I'm happy to take questions depending on whether or not we're still on time. If not, I'll be hanging around after the call and can take questions there. So thank you all so much.
Thank you. I think what we wanted to do was if there was a pressing question that came up from the audience in the chat, I don't see one currently, we would take it, but otherwise we'll take questions at the end. So
there are a couple of questions, Damien, that are there, but there was a question from Divya Varadarayan.
Okay.
In network analysis results where you compared fetal with adults, there were some connections that were stronger in fetal than adults, more in the cross-term regions, not near the diagonal. What are those?
Good question. So we, as a group ourselves, have not compared adults to fetals. We typically do either preterms or neonates as our control group. Prior is a little bit more common to do adults. I think there are a lot of concerns doing it that way. You don't see it as much anymore because there is so much development that occurs between the fetal period and adulthood, where the brain is really specializing into subnetworks, which is not as common earlier during fetal development. That study also was collapsing across the fetal period from 20 weeks to 40 weeks, which is not something we do quite as much anymore because of how much we know the brain changes during that period. So I would have to reference the study itself to see exactly what those were. Yes.
Thank you. There was also a question about how much, when you do resting state data, how much do you actually acquire in order to be able to analyze? What's the lowest limit of resting state data that you need to acquire to be able to analyze it?
Yes, so we still largely work at 4 minutes of usable data. It's a little low. I don't like doing it as much from what we know from adults and adolescents. But in order to get 4 minutes, we still sometimes collect 12, 15 minutes of rest just to get there because motion is so severe. So it varies. I am pushing more towards at least 5 minutes. I would love more, but as you saw, especially at younger gestational ages, those kids are just going to wiggle around. Once we get closer to term age and they get restricted by that uterine environment, we don't need to worry as much because there's that natural packing keeping them in place. So it largely depends on the age range we're looking at, but we lose probably about half of our scans total to motion, even trying to keep 4 to 5 minutes of rest.
The others can kick in if they have other ideas to these questions. I think that's the idea of our panel. There was a third question that was really wanting to know what these fetal functional connectivity acquisitions are. Are they 2D? Are they truly 3D? And how would you address motion within a volume that you acquire?
So they are 3D acquisitions and motion. So I talked a little bit about how we address motion. Outside of what I presented today, we pretty much mirror what is done in ex utero work. So if I didn't mention a deviation, it's because it's pretty standard to what we're doing at any age. We just have those additional steps to deal with the fact that we have such high amounts of motion.
So perhaps I can add, Petra. So I'll talk a little bit about the fetal acquisition in the fetal DHCP, but we've always used a 2D slices, and what we do is then do a slice to volume reconstruction to make a 3D volume. And that allows us then to have a bit of redundancy for motion, because of what we do with the slice to volume reconstruction means that also we do ascending. We used a little bit multiband, but we tend to use traditionally an ascending acquisition, which means that we can then account for what happens with signal evolution from slice crosstalk, and then the longer T1 means that we've got longer spin history effects. So we can do some I suppose, more effective control for those spin history effects by using a 2D acquisition.
So you're getting challenged in the chat. 4 minutes. Ravi Menon thought this was like really crazy short. He says 10 to 120 minutes, but then of course we are challenged as
Yeah.
Wow, 120 is a bit enthusiastic.
Yeah.
We do around 10 minutes.
Yeah, but this seems like a perfect topic for like having larger discussion with the group to make sure we stay in time. Probably it's good to move on to the next speaker.
That sounds good. So do you want me to introduce Chris Smyser? I can certainly do that with great pleasure, right? Chris Smyser, professor in pediatric neurology at the Washington University in St. Louis, and head of the one of the big developing brain imaging groups. And we're looking forward to hearing his view on preterm brain, I would assume. Thanks, Chris.
Yep, thanks, Petra. Can you guys see my slides okay?
Yes.
Perfect. Okay, so as Petra just alluded to, excited to be with you here this afternoon, to talk about some of our group's work in the prematurity space. This will hopefully segue nicely between Kevin's talk and Tom's talk, and I'll try and lean into some of the methodological questions that we've already alluded to and how that permeates across studies and populations. Okay. So as this group is well aware, premature birth is a major public health issue. In the United States alone, more than 500,000 infants are born prematurely each year. And despite significant advances in this area of medicine, there's really been no appreciable improvements in rates of preterm delivery over the course of the last decade. During this period, we've also become increasingly aware of the long-term challenges faced by surviving infants. So those infants that are born earliest are at greatest risk. 10 to 15% will develop cerebral palsy or some sort of motor impairment. More than half will have some sort of cognitive or learning challenge. And at least 25% will have some sort of behavioral problems. So anxiety, ADHD, or autism, what we collectively refer to as the preterm behavioral phenotype. In the United States alone, the estimated annual cost to care for this clinical population is more than $26 billion and growing. So with that context in the background, I'd like to introduce you to a patient of mine who's helped shape a lot of what I do in my research in this space. So this was a patient I cared for in the St. Louis Children's Hospital NICU. This is a picture of her when she was just a few weeks old. You can see that she's intubated, has eye protection, and is in an incubator. She was born at 26 weeks gestation. Her pregnancy was complicated by being a twin. Mom was actually a NICU nurse, went into spontaneous preterm labor, which we were not able to get stopped, and she was ultimately delivered via C-section due to breech presentation. You can see here she had some initial challenges in the delivery room reflected in these APGAR scores, which are scaled at 0 to 10. She was intubated in the delivery room and then transported to the Children's Hospital NICU for further evaluation and management. And so it was there that she underwent this head ultrasound study on day of life number 3. Not sure how many in the audience are familiar with looking at neonatal head ultrasound studies, but this is the anterior fontanelle up here. This is essentially a coronal image at a little bit of an axis, the inner hemispheric fissure right here, left hemisphere, right hemisphere. In these images, the spinal fluid is hypointense. So this is that left lateral ventricle, choroid plexus with perhaps a little bit of hemorrhages here on that left side. In contrast, on the right side, we could see that right lateral ventricle is nearly entirely filled with blood, which is very hyperintense. And then further still, this particular baby had experienced hemorrhage into the surrounding parenchyma or brain tissue, so a periventricular hemorrhagic infarction, or what historically has been called a grade 4 IVH. So she then went on to have a 3-month NICU course complicated by many of the common comorbidities of prematurity. So she had a patent ductus arteriosus, hyperbilirubinemia, chronic lung disease, apnea prematurity, and some challenges with feeding. And then as she was getting ready to go home from the hospital, we obtained this MRI scan at term equivalent age. And so again, these are T2 weighted images, as we could tell from the bright spinal fluid, axial images here on the left, coronal images here on the right. Again, just to get you oriented in terms of what we're seeing in this left hemisphere on the axial image, a nice cortical ribbon around the outside of the brain, confluent unmyelinated white matter. And then we can see that left lateral ventricle with the spinal fluid being bright, maybe a resolving hematoma on that left side. But for the most part, the left hemisphere looks relatively normal. Contrast that with what we see in that right hemisphere, with that right lateral ventricle has a large amount of clot burden still being broken down, that hypointense blood, and then the degrading blood through here. We flip over to that coronal image, and we can see, again, a left hemisphere that looks relatively normal in terms of its appearance. But in that right hemisphere, we could see this already emerging porencephalic cyst with a clot that is being degraded in that right lateral ventricle. And so as a clinician, these lead to a common set of questions that are really core to our mission. And, in a big picture sense, it's always, what do we tell the family? What does the structural MRI scan tell us about both brain development? And then perhaps most importantly, what does that indicate for long-term outcome? Are there other studies we should think about obtaining, and what should be the timing of those, if so? And are there specific clinical interventions that we should consider? And if so, when should those be discussed with the family and potentially provided? So there was really a lack of data at the time regarding the answers to these specific questions and how we could contextualize MRI into conversations with families at the bedside. And so that was what led our group at the time to explore what advanced neuroimaging modalities could provide in terms of these conversations. And so that was really now 10 to 15 years ago where we started delving into techniques like resting state fMRI or functional connectivity MRI, which, as this group is likely full aware of, measures the temporal correlations and low frequency fluctuations in blood oxygen level dependent signal that occur independent of task, similar to what Kevin just discussed. And we can use these data to identify networks which demonstrate synchronous spontaneous neuronal activity. So just leaning in a little bit more, I like to use this simple cartoon to depict some of the concepts that Kevin discussed and that we'll be focusing on in the remainder of the talk here. So as we think about the brain being made up of a series of independent regions, each with their own spontaneous patterns of blood flow, we can pick a region or a series of regions and look at the blood flow patterns there and look at how those relate to other areas throughout the brain. And in doing so, we can identify these synchronous spontaneous neuronal activations, or these resting state networks, as you can see here depicted in yellow. So it was based upon this premise that investigations in adults and then later in older pediatric populations consistently identified these resting state networks located throughout the brain. And what you can see here are figures that are likely very familiar to you, depictions of 6 well-known canonical resting state networks, including those that are located in higher order association cortices like the default mode, frontal-parietal control, and dorsal attention networks, and contrasting those with networks that we see in those earlier developing sensory motor association, or sensory motor cortices, the visual, motor, and auditory networks. And so our group was one of the early investigators of whether these networks or their precursors were present both in healthy term-born infants, as you can see depicted here, and then further still in very preterm infants, those born prior to 32 or 30 weeks gestation, like the infant that we started with in the introduction to the talk here. And so as we consider investigations in this population, kind of building again on what Kevin just provided in his fetal discussion at the macroscopic level, it is important to acknowledge the relatively massive amount of structural change that is occurring during this critical developmental period as a result of typical brain development. And so what you can see here is work by 2 talented former grad students in the lab, Jason Hill and Kara Garcia, which demonstrate the changes the brain is undergoing during the period of time when infants are cared for in the NICU. And so, again, these are results from babies from the NICU at St. Louis Children's Hospital, demonstrating how the brain changes from 27 to 30 to 34 and finally 38 weeks post-menstrual age. And of course, we can see how much the brain increases in size, but we can also appreciate how much folding occurs during this period. And this is really critical because your folding pattern at term equivalent age is highly correlated with that into adulthood. And so what this tells us is that if your brain does not develop typically during this window, that it has potentially long-lasting effects. And so again, just to familiarize yourself with some of the procedures that we use, again, many of these might be ones that you use at your centers to collect infant functional or just standard MRI data. So we developed a series of procedures which allowed us to standardly acquire data without the use of sedating medications, which was the key factor at that point in time. So you can see hearing protection, receiving blankets, a vacuum fix device, which has almost a beanbag sort of consistency to it. We put the babies in, pull the air out, form a papoose-like structure into the head coil, and then on into the MRI scanner. And with greater than 90% success, babies will sleep through the MRI scans. And we've done this for thousands of scans at our center with no complications. And again, most importantly, these procedures can be readily employed at any institution. More recently, over the course of the last several years, we've incorporated the use of FIRM, which is an innovative MATLAB-based algorithm which provides real-time feedback at the scanner console to the scan operator regarding the amount of low motion data you're acquiring during an individual session. So here's a representative tracing where for the most part, the baby remains still, and we can see these very brief epochs where there's high amplitude head movements. And then again, this is broken down in a display that's available at the scanner console. So really feeds into the concept that we were just referencing regarding how much data you're collecting and the importance of that in terms of our analyses. So working with Andrew Degnan, now a pediatric radiologist, what we found was that we were able to identify multiple resting state networks located throughout the brain in our term and prematurely born infants. These were located in cortical, subcortical, and cerebellar areas. You'll see I like to use pictures to depict key results. At the time, we were using seed correlation analyses in our early approaches, and you can see we investigated a series of networks shown by the regions of interest across the top row. And then here's results for healthy term-born control, and then term equivalent, very preterm infants. So infants scanned at comparable postmenstrual ages. And again, you can see these networks located throughout the brain. Interestingly, when we started to compare these results to those from adults or older pediatric populations, we saw that there was a lot of similarities. And this was particularly notable for networks located in the primary motor and sensory network. So again, these are mean correlation maps that are generated using a left motor cortex seed. And in these results, you can see positive correlations denoted in warm colors, negative correlations denoted in cool colors. Again, representative mean correlation results for the adults, our term control, very healthy term-born infants, and then our term equivalent, very preterm infants here. And again, just simply working your way from top down to bottom, you can appreciate there are a lot of similarities. This was particularly true for those networks located in the sensory motor cortices, but perhaps less so for those located in higher order association cortices or networks. We pursued similar investigations in the cerebellum. So this is work from Charlotte Hersman, a former postdoc in the lab, which demonstrated these same types of patterns were evident when we explored the patterns of functional connectivity between the cerebellum and the cortex. The cerebellum, of course, is a structure known to be critical for early brain development and plays a key role in many developmental functions. And again, we found comparable results in both infants and adults using a winner-take-all approach, as you could see illustrated here. We also sought to investigate how these networks change during the course of early development. This is akin to what Kevin was just discussing, looking at some of the longitudinal fetal data. And what we found that were these individual networks demonstrated age and network-dependent patterns of maturation. And these results are depicted in this figure here. Just to, again, get you oriented, in the furthest left column are the seed regions we used for our seed correlation analysis. And then as we worked our way from left to right, we can see the mean network results at 27, 30, 34, and 38 weeks postmenstrual age. And again, results for our healthy term-born control infants shown on the far right. And you can appreciate that the majority of these networks change gradually over the course of early development while these infants are being cared for in the neonatal intensive care unit, with the majority of networks consistently demonstrated correlations between homotopic counterparts. We also observed that areas known to demonstrate early cortical development histologically are also the areas where we see mature resting state networks emerge first. Next, working with Tyler Blasey, a current postdoc here at WashU, we investigated resting state networks in very preterm infants with forms of white matter injury common in this population. So diagnoses like cystic PVL, IVH, and post-hemorrhagic hydrocephalus. And what we saw, perhaps not surprisingly, was that results in this population demonstrated significant alterations in infants with cerebral injury in a pattern that was dependent upon both the proximity to the injury site and the severity of injury. Importantly, and I think interestingly, the areas of injured tissue were not incorporated into the resting state network. So again, a nice example from a paper that Tyler contributed to. Our healthy term-born control results in a representative infant, our term equivalent very preterm infants, and then infants with different severities of brain injury. And again, the main takeaway here is as you work your way from top to bottom, we can see the effects of prematurity and then increasingly severe forms of brain injury and how that's reflected in different resting state networks throughout the brain. We also performed a series of investigations, specifically investigating the effects of prematurity alone, intentionally excluding those infants with brain injury from the analyses. And I'll talk through a few different ways that we've approached this type of question and then how we have started to think about relating these imaging measures to outcomes. But we started simply looking at a network by network basis. And so again, this was an investigation that we performed looking at thalamocortical connectivity. We used a left thalamic seed here. We looked at the correlations between the thalamus and then extending superiorly up into the sensory motor cortex and then inferiorly down into the brainstem and cerebellum. These are results from our term equivalent very preterm infants. Identical analyses performed in our healthy term-born control infants. Again, qualitatively, you can appreciate that there are differences throughout the brain in these two sets of maps. And then what you see on the far right are the results from a direct statistical comparison where those voxels that are colored red were those that were statistically significantly different between the two groups. We expanded our quantitative group comparisons, looking at correlations within and between the canonical resting state networks that we discussed earlier. So what you can see here are a series of mean correlation maps generated using 31 regions of interest from 7 resting state networks located throughout the brain. These are the mean correlation results for our healthy term-born control infants, shown here on the left, our term equivalent for preterm infants. So again, subjects scanned at the same age. Similar to before, qualitatively, we can appreciate differences throughout the brain between these two results. And then on the far right, we can see the results from the statistical comparison. I'm not sure how well it projects on your screen, but every cell that is denoted by a black star was, again, statistically significantly different between the groups. So effects not localized to one specific network or one specific area of the brain, but really truly brain-wide differences between these two populations. We leaned into these matrix-based comparisons, and working with Nico Dosenbach, a colleague here at WashU, we employed support vector machine multivariate pattern analysis to investigate these differences further. In order to have a robust data set to do these sorts of analyses, we placed regions of interest in cortical and subcortical regions throughout the brain. And in doing so, we use these ROIs to create significantly expanded correlation matrices. Similar to those described previously, what you see here are results from 214 regions of interest generating correlation matrices with nearly 23,000 functional connections between ROIs for each individual subjects. And then by concatenating these matrices across all subjects in the cohort, we were able to create data sets of sufficient size to appropriately apply these mathematical approaches. And what we found was that with approximately 85% accuracy, we were able to differentiate between our prematurely and term-born infants scanned at comparable postmenstrual age. We could extend these results to identify the functional connections that were specifically important for differentiating between the two groups. And you can see those results here. These are weighted based on the relative importance of each connection. Those that are colored in green were those that were significantly stronger in our term control infants. Those that are colored orange were significantly stronger in our very preterm infants. And then finally, we could extend these results to support vector regression, training an SVR model to predict gestational age at birth based upon an individual infant's resting state data set collected at term equivalent age. And so in this set of results here, the actual gestational age of the infant at time of birth is shown along the x-axis. The predicted gestational age based on their individual data set is shown along the y-axis. And again, we can see that the model did a nice job of differentiating between our healthy term control and very preterm infants scanned at comparable postmenstrual age. So segueing into some of the work that we've done looking at brain and behavior, through a great deal of effort from our lab staff, we have been able to bring back more than 80% of our preterm cohort for standardized developmental assessments at multiple time points during early development. And we've used these behavioral data to explore brain behavior relationships between our neonatal functional MRI results and then childhood outcomes in our injured and uninjured preterm samples. And here's an example from Petra, a former MD PhD student in the lab who's now doing a PM&R residency, who investigated how well neonatal functional connectivity results for the motor network extending into the basal ganglia and cerebellar regions related to motor performance during early childhood in our preterm cohort. And shown here are data that demonstrate the strong relationships between motor network correlations shown along the x-axis in these plots, and then behavioral outcomes, in this example at age 2 years for Bailey assessments of motor performance along the y-axis. You can see the breakdown here for our injured infants in red and our uninjured infants in black. And what you can see at the relationships are much stronger in the injured infants in this particular analysis. We see similar results in a paper that was recently published at 5 years of age, again, with Petra as the first author. As I mentioned previously, these infants are at high risk for psychopathology. So through correlations with Cynthia Rogers and Chad Sylvester, two of my colleagues here in child psychiatry at WashU, we've also explored the relationships between neonatal functional connectivity and early psychiatric outcomes, assessed using a series of different tools at 2 and at 5 years of age. And we found that the strength of neonatal functional connectivity predicted both internalizing symptoms and behavioral inhibition at ages 2 and 5. Importantly, these analyses identified regions of the brain previously identified to play a critical role in psychiatric disease in older pediatric and adult populations. So again, what we have here is a nice example of neonatal results being associated with early childhood outcomes, this time in a totally different developmental domain. We've continued to follow these children and talk a little bit about these longitudinal investigations in the next couple slides here, bringing them back at 9 and 10 and putting them back in the MRI scanner and looking at how the brain changes over the course of time. This provides us with a unique opportunity to interrogate how their brains have changed over the first decade of life. And so here are a set of results from Lisa Gorham, an MD PhD student in the lab who has studied cortical expansion between the neonatal and 9-10 time points using a modeling approach called multimodal surface matching, or MSM. And the top images here, warm colors denote areas of greatest cortical expansion from birth to 9-10, and those that are cooler colors denote less. And bringing this back to the functional connectivity analyses that we're talking about here, in this bottom image, we've denoted with black lines the margins of the DMN on a group average expansion map. And so really the takeaway here is that there are areas of significant overlap between this network and areas of high cortical expansion at this time point. And what this tells us is that there's likely to be direct intersects between what we're seeing from a macrostructural and a functional perspective as we look at longitudinal brain change over the course of time. And finally, more recently, working with Mariah Wheelock, one of our faculty members in radiology here, we've examined brain behavior relationship between functional connectivity and standardized motor performance using a mathematical technique called enrichment analysis, which we've adapted from the genetics literature. I won't delve too deeply into the technical details here, but really the main premise of this technique is that we look to leverage brain-wide functional connectivity data rather than limiting ourselves to look at specific networks or regions of the brain alone. And what we found using this technique was that term and preterm children rely upon different networks to perform the same types of motor activities at specific time points. So here are some of the most prominent differences in terms of these results that we identified in this population. And what you can see here in this top set of images was that our term control children relied heavily on thalamocortical connectivity to perform motor tasks, which we would typically predict to be the case. And in contrast, if we pivot to these results for the very preterm populations, we found that they relied much more heavily on the cerebellum to complete these same tasks. So what this suggests is that there might be potentially compensatory alterations in terms of brain development, in terms of some of the brain behavior and how these change or evolve over the course of time. We did a similar line of investigation looking at measures of attention and found similar patterns where there were fundamental differences in the networks involved in tasks between our very preterm and term control populations. And so what are we pursuing with this cohort now? Well, in collaboration with Cynthia Rogers, who I co-direct the lab with, we are very excited that we're bringing these children back at age 14, 15 for the next round of comprehensive assessments. And this time we're really focusing or leaning into the role of the cerebellum and its prominent presence in terms of some of the brain behavior findings that we've already discussed over the course of the early time points in this particular high-risk population. So again, here's some of the early results where we're seeing in this image here, the differences where there's stronger corticocerebellar connectivity in our healthy term control participants and compared to our very preterm children. And then what we're seeing here are, again, some fundamental differences in terms of corticocerebellar activity as it relates to some of the measures of social competence and deficits that we're seeing in our preterm subjects in that population. So hopefully much more to report in the coming years regarding the results at this time point, as well as we continue to study these children longitudinally. And so in the last few slides here, I just wanted to pivot very briefly and hopefully provide a nice segue to some of the work that Tom's going to discuss. And I just really want to hearken back to the point that we were just discussing about the importance of methods and just thinking through and evolving with the field as change continues to permeate throughout the analyses that we're doing with time. Kevin alluded to this, and I think this is really important and really just a fundamental aspect of the team science that we're all keenly interested and motivated by here. So I just wanted to touch on some of the work that Chad Sylvester has done more recently, looking at some of the different analytic techniques where we've begun to map our neonatal data onto the surface in terms of some of the analyses that we're doing. Look at the amounts of data that are necessary, leaning into the fact that we might benefit from having prolonged acquisitions. There's also a series of parcellation schema that are available from our group and others, just looking at the different ways that we can map functional connectivity in neonatal populations and do so in an increasingly intentional fashion. I think some of the work in this space has really been motivated by some of the large-scale data sets like the DHCP cohort that Tom is going to talk about, and then the HBCD cohort, which Damien hopefully is going to touch on briefly in some of the discussion points as well. And this is really key because what we are increasingly appreciating is the more we refine methods, the more we collaborate, the more we work together as a field, the more similar our results in neonates appear to those from adults. So again, this is some of the work that Chad has done, really leaning into some of these optimization of the different analysis techniques and the ways that it reflects patterns in the association cortices in particular that look very adult-like when we continue to fine-tune things. And then work from Ashley Nielsen from our group, which again, just drives home the point that employing these techniques can really refine the way that we think about functional system development in the early aspects of brain development that we want to study in preterm and healthy term control populations and those at different types of medical and clinical risk. And so to tie everything back together in the last couple of minutes here, I hope I've demonstrated that resting state fMRI provides an important tool for assessment of functional brain development in normal and high-risk neonatal populations. We and other groups have been able to successfully identify networks located throughout the brain in our term and preterm cohorts in a manner that reflects what we know to be true about cortical histology. There are many different techniques or approaches that we can employ to examine group and individual level differences across cohorts. And again, very excited about the fact that we're now linking early brain to subsequent behavioral outcomes into early childhood and then now ultimately into adolescence and beyond. So just to try and tie everything back together, the patient we started with, so she's now 15. We put her back in the MRI scanner. We brought her back for standardized developmental assessment as part of the longitudinal cohort. So she's now in the 10th grade or high school. She's in a regular classroom. She has a mild left hemiparesis that's most prominent in her distal left lower extremity. It's one that really the neurologist is the one who's going to pick out, not somebody that her peers would notice as having any sort of motor or movement problem. She does have some anxiety in new social environments. She continues to receive therapy. She has some orthotics that she wears in her shoes, but she's very active, part of her local community, runs cross country on the high school team, swims with peers. She works as a lifeguard. Here's what her brain looked like when we put her back in the MRI scanner at age 15. So T1 weighted images. This time you can see axial at the top, coronal at the bottom. Again, we can see that this poor encephalic cyst has continued to evolve since we saw the brain pictures last. We can do detailed segmentations of the different tissues in the brain. We can look at the microstructure of the brain with these tractography images and appreciate the significant impact of the cortical spinal tract from that poor encephalic cyst and that early brain injury. And we can begin to look at the different patterns that we can see in terms of functional brain development as well. So very excited about looking at these types of cohorts with longitudinal studies over the course of time. And so with that, obviously, a big group of folks who are integral to all of the work that we do, and obviously very important to acknowledge families who continue to come back now for many years and continue to participate in our studies and our funding sources as well, and happily take questions now or at the end of the session. But thanks very much for your attention. Again, thanks to the organizers for bringing us all together here. Much appreciated.
Thanks very much, Chris, giving a really medical perspective of all the imaging questions that are certainly uprising in the ISMRM community. Damien, do you want to lead the questions, or there's not much in the chat right now?
Yes, I think there's the one question that did pop up that I think maybe you can help answer, Chris, is there a movement towards precision fMRI in infants?
Yeah, so the short answer is yes. I think it's something that there are a lot of investigators keen to explore. I know Chad Sylvester, whose work I've obviously referenced already, is working with folks at the University of Minnesota with Damien's team, and I know there's other groups working on this actively as well. It's obviously complicated because we need to be intentional or thoughtful about how we put babies in the MRI scanner for longer acquisitions and how we do that serially over the course of time. But I think there is a movement right now underfoot to try and think through how we leverage the longer acquisitions and what those afford, and then what we can continue to extract from shorter acquisitions and how we can work essentially bidirectionally in that space and use priors to inform things, use the data and the outputs from analyses from the longer acquisitions to inform what we might be able to do for clinical populations in that translational piece as well.
Yeah, thank you. And it's interesting too. There's a paper that just came out by Thomas Yo in Adolescents and Adults, showed, which essentially showed the economics of longer scans depending on the types of questions that you ask. You know, so if you're asking questions about within individuals, like the longitudinal data that Chris showed, it's a different proposition if you're doing than if you're doing like an association study with some outside measurement, you know, that's kind of across people. Probably a good read. It's almost certainly going to be very different in infants. But the thought process is good for how we're designing experiments. So that's good. All right, Tom, you're up. I think you're going to be taking us home here talking about DHCP.
Thank you, Damien, and thank you very much, everyone, for organising this and attending this session. It's really exciting, actually, to be invited and to talk to the brain function group about it. So hopefully we can raise some interest as well in the field. It's something obviously Chris and Kevin and I are very passionate about, and Petra and Damien. So it's great to have it discussed with a wider brain function group. I may have too many slides, by the way, Damien and Petra, so do cut in if I'm overrunning.
Okay.
All right. Can you guys see that?
Speak so fast, you will manage.
Can you guys see that?
Mm-hmm.
Great. Okay. Thank you so much. So I'm going to try and briefly run through a little bit about the DHCP and explain, I suppose, more of a kind of technical perspective about how we try to overcome some of the challenges inherent to imaging fetals and neonates to try and get good functional data, to introduce the data set and then hopefully enable you guys to then think about how you can use that data. Chris has already very eloquently talked about the kind of differences that you can see in preterm infants, and then Kevin, of course, has talked very beautifully about what you can see in the functional fetal data. So I thought I probably wouldn't touch on too much about what can be done with the data in terms of looking at network structure and so on. And I was then going to spend the second half talking a little bit more about some of the work that we've been doing to try and understand effectively what actually is the bold signal, and understanding how it can really tell us a bit more about brain development and how we can try and unpick some of those things that underlie the bold signal that we're actually looking at this stage. So first of all, I was just going to very briefly, and actually since Chris and Kevin have done such a great job, I probably won't go into in too much detail about what fMRI can tell us about functional brain development in early life, what some of the challenges are really inherent to imaging this population. And then I was going to introduce the DHCP to talk about the different kinds of approaches we took to acquisition and processing in the different populations. And then lastly, as I said, to introduce how we've tried to understand BOLD in the developing brain, with a particular focus on some work we've done with simultaneous EEG fMRI, and also more recently with ultra high field fMRI at 7T. So as Kevin really showed very beautifully, the brain is changing in an enormous way at this time, with the cortical folding coming across in addition to the growth. So we can see that the cortex and its surface area actually increases at a much higher rate than the size of the brain and the volume of the brain itself. So underlying this, of course, there are dramatic changes, what's happening in terms of the function of the brain and the way the kind of different patterns of activity are laid out. So on the left, we've just got some resting state networks, again, showing the kind of things that Kevin and Chris have talked about, where we can see a change from a network which is located predominantly on one side of the brain in the earlier stages. And then you can see as we start to reach the older ages that we start to then get the homologue on the other side, so we start to get a more established bilateral resting state network, similar to those seen in adults when we get to term equivalent age. On the right side of the screen is some work that we did with task-based fMRI. So this is a movement of the right wrist. And what you can see is that the infants had a contralateral activation pattern at the youngest age in the preterm kids. And then as they get older, you start to see more elaboration of that response, with the ipsilateral hemisphere involved in the integration of activity in the thalamus, and also you can see the supplementary motor area, the midline, coming up as well. So together, then, this is just really to summarize what really the field has kind of shown so far. And I'd just like to hasten to add that although I've made this figure quite recently, as Chris mentioned, one of the things that's really very clear is that as we start to improve our methodologies, we improve our sensitivities, and we try to improve the way that we can actually image these different networks and characterize these networks. It's probably going to change this kind of summary slide. But at the moment, what we can see is that resting state networks, we can certainly see them from the start of the third trimester, and they predominantly start as areas of local connectivity in one hemisphere. As time progresses, we start to see long-range patterns of connectivity being established, especially including the interhemispheric connections, first of all, across the third trimester. And then longer-range patterns of connectivity are first established in the primary sensory and motor networks, and then connectivity is established later in the high-order and associative networks. And I think these third and fourth point, I think it would be very interesting to really see what happens when we start to do this kind of precision mapping and so on, that we can start to understand with longer acquisitions and better characterizations, we might improve our sensitivity. We might understand a little bit better when these things are happening, but also precisely whether or not they really are something that happened later on in infancy, or whether or not it's just that we lack sensitivity to be able to recognize them earlier. And last of all, we can also even see complex network features such as gradient organization, which is something that's happened when people have really dug deeper into our DHCP data. So some of the challenges which Kevin's touched on and Chris have touched on are very important to consider when we think about doing functional fMRI studies in this age group. And that's because, of course, imaging these infants is not just a case of using the same methodologies that was used in adults. It's not just a small adult brain. There are lots of changes happening across that time. So in addition to the changes in kind of configuration of the cortex, the shape of the brain, of course, the tissue composition is changing very dramatically as well. There's much more water in the brain at the start, and then as time progresses, you're going to see big changes. I mean, on across to the top here is just a T2 atlas, but you can see that there are big changes, particularly in the white matter in terms of the signal intensity. And as the tissue composition changes, then of course we see significant changes in our tissue-specific parameters like T1 and T2, and in particular T2 star. And so, of course, what we traditionally would try and do with fMRI acquisitions is to match our echo time to the T2 star of that particular tissue. And if we consider that T2 star times, because of the amount of water in the brain and the kind of composition of the cortex is changing, then what we see is that optimal T2 star times, or sorry, T2 star times, and therefore the optimal echo time that we should be acquiring our data with is gradually but significantly decreasing with age. There's also changes in brain size and anatomy, which of course have very important implications for effective resolution and how precisely we can delineate what's happening in the cortex. And then there are, of course, developmental differences in cerebrovascular physiology as well. And that directly has implications for the neurovascular coupling, which we assume is underlying the kind of bold responses that we look at. So this is quite old work that I did many years ago, just showing that if you were to look at the induced bold response to a very simple somatosensory stimulus, that the youngest infants shown here in green have a delayed and lower amplitude bold response compared to the traditional canonical adult HRF, which you can see here in blue, with the response happening much faster and much higher amplitude. The other major issue, which Kevin talked quite a lot about, is uncontrolled head motion, which can cause marked significant artifacts. Not just the signal itself, but can also cause changes in the kind of distortion effects that we see in the interaction with the surrounding maternal tissue. And then the last one is a much trickier thing to try and think about, but of course there are going to be massive differences in the observation mode itself. So if we think about how we're trying to image a fetus, with usually some kind of body coil or some kind of cardiac coil that's put on the maternal abdomen, as opposed to a neonate where we've got them, depending upon whether or not you've got a specific neonatal coil, you may be using an adult coil, the position of the coil elements, the received coil elements, is going to be completely different. So that in effect is also going to affect your SNR. It's going to cause different kinds of signaling heterogeneities. I'm talking not fast enough, Petra. My slides are moving faster than me. And that can then, of course, cause you different kinds of sources and different characteristics in the potential noise in your sensitivity. So just briefly talking about the DHCP. So we collected this data from 2014 to 2019. It was a project which is funded by the European Research Council, and we conducted it in collaboration with Imperial College London and Oxford University, although the data itself was all collected in a single institution here at King's College. The overarching aim of the project was to create a dynamic map of human brain connectivity from 20 weeks post-conceptual age all the way through to 44 weeks. And we wanted to really link together not just the imaging, but also detailed clinical and behavioral and genetic information as well. And in doing so, then we wanted to create an open data resource for obviously the wider research community, the neuroscience community, and also really for people to start probing it for clinical reasons as well. And one thing that we were really very clear about when we set out on the project is that we really wanted to start right from square one and develop specific acquisition methods and processing methods and analysis methods really specific for this population, so we could get the highest quality images that we possibly could. So the data set itself consists of 1,228 data sets from 1,173 individuals. So the reason those numbers are different, obviously, is we have a few who have got longitudinal data where they were imaged both as fetuses and then again as neonates. The majority of participants were healthy infants delivered at full term, although we did enrich our cohort with about 200 preterm infants and a further, I think it's around 200 infants with a family history of neurodevelopmental conditions like autism. There's detailed clinical and sociodemographic data, and we have developmental outcomes which were collected at 18 months. So you can see on the bottom right here a list of those outcome scores that were taken through questionnaires and different child assessments from the infants. Unfortunately, the COVID pandemic kicked in towards the end of the project, which is why you can see our numbers are around 70% for the developmental outcomes, and that's because unfortunately we were unable to invite back a significant proportion at the end when they were due for their follow-up. They also had eye tracking assessments, and a subset again has had genotyping and DNA methylation studies as well. All of the data is free to download. It's available. I've put the link here through the NIH portal, and there are also links in the various papers that have come out of the work to our different tools that we've developed on GitHub. So as I mentioned, one of the things that was very important for us was to really think about how we can acquire the best data possible and to really think about all of the different stages of the acquisition. So one of the first things that we did was we spent a considerable amount of time thinking
Did we lose Tom?
I think so.
I don't know. I sent him a chat message, but maybe I need to send him a message on the.
which we then developed with.
Okay, Tom, we just missed you for a little bit, so you might have to rewind on that slide.
Oh, sorry. What point did you lose me, Petra?
Just when you started on the dedicated neonatal brain scanner.
Oh, okay. So we wanted to develop a system that would allow us to be able to acquire the best possible images with a coil which was sized for the 98th centile of the neonatal head. And then also you can see here that we developed a specific shell in which the infants can be positioned outside the scanner and then easily lifted into the coil for imaging. And you can see here the shell and the system positioned in addition to an acoustic hood to try and decrease vibrations and sound during image acquisition in our Philips 3T scanner that was used for the DHCP. Inside, this is an infant being placed into that shell, and inside that we then use inflatable cushions that were developed with Pearltech, which help to position the infants by being inflated around the infant head, but also help with acoustic noise. So they act, instead of having although here you can see there are mini masks, we actually discovered that we didn't need the mini mask because the inflatable cushions were in effect acting as acoustic protection. So just moving on now, I'm not talking at all actually about the structural acquisition. So what we did is we imaged the infant for 60 minutes during natural sleep on the 3T Philips system. And we divided it, broadly speaking, into 20 minutes of anatomical imaging, 20 minutes of diffusion imaging, 20 minutes of fMRI acquisition. So I'm just going to talk about the fMRI acquisition. Within the 20 minutes, we acquire data self, but also complementary data for distortion correction and for the processing pipeline. One of the first things that we did when we begun during the preparation was to try and think about the optimal echo time, because as I mentioned, we would anticipated the echo time required would have to be longer than conventional adult acquisitions. And so when we looked at the sensitivity, we effectively found, looking at group ICA for the required contrast to noise and for the kind of T statistics for resting state networks, that traditional kind of echo time that might be used in an adult acquisition of around 25 to 30 seconds was not appropriate. But once we moved up to around 45 to 65, you can see it starts to level off there before it starts to deteriorate when you move much higher. And so what it suggested is we did need to use a longer echo time than the normal 25 milliseconds. The second of all, what we wanted to do is to try and think about our optimal temporal resolution, so our sampling scheme, so that we could avoid physiological noise. The neonatal heart rate is much, much faster than an adult heart rate. So neonates, their hearts are beating around 120 to 160 beats per minute. And so there is a real danger that if we acquire it with a more traditional TR of kind of 1.5 to 2 seconds, that what you might do is, and I'm afraid it's tiny on here, I'm aware of that, but maybe you can see my my arrow, you would aliase in then the cardiac and the respiratory noise into our sampling. So it would be folded into the fMRI data itself. And so what we did then is we really thought about how we can move our acquisition to a much faster sampling time so that we can try and separate out or move away from having those physiological noise sources folded into our acquisition, and they're separate. And that means then they can be precisely denoised from the data to give us more reliable functional connectivity estimations. And so this was the final acquisition that we then resulted in. So we have 15 minutes of fMRI data that's collected with an echo time of 38 milliseconds, a TR of 392 milliseconds, which we achieved with a multiband factor of 9. And we collect 2,300 volumes with a resolution of 2.15 millimeters isotropic. The release data includes both the unprocessed reconstructed data and then also the processed data. And then we've also included the different registered data and the respective transforms to template space. So here, just to give you an idea about what the pipeline involves. So this pipeline, again, is available on GitHub, and there's a very detailed paper from Sean Fitzgibbon, which was published now a while ago in Neuroimage in 2020. So the raw data you can see on the left here, and you can, of course, see here that there are some slice effects, both through the multiband acquisition, but also because of motion during the acquisition. So we then do a slice to volume motion reconstruction to produce a much nicer 3D volume that you can see here. And to align our slices, and you can see that there's still some multiband artifacts within that. We then do a dynamic susceptibility distortion correction. There's a robust multimodal registration then incorporated into that, and then we use ICA denoising, which you can see on the right here, so that we can try and define areas of signal, but then also a multiband artifact, different kinds of sources of movement artifact, physiological artifacts such as cardiovascular noise, both in the arteries and the sagittal sinus, and then various other sources of noise, which are then removed and denoised. And then we have an automated QC framework, and the results of that automated QC framework are also available for each subject when people download the data. And that means eventually then that we could see we've got our raw data here, and on the bottom here, the temporal SNR for the denoised data, where you can see a dramatic improvement, particularly for the cortex around the brain. Just touching very, very briefly on the kind of insights then that can be derived from the data. So this was work from Mike Eyre now several years ago, just showing that then having this kind of data, both the quality of the data and also the amount of data, means that we can look across 400 infants around term age. So these are all infants who are born at term. And you can see that even across this short period of what we'd consider to be full term from 38 to 42 weeks, so across this 5 weeks, that there are significant differences in terms of the kind of strength of the connectivity from one side of the hemispheres to the other. Even these kind of resting state networks, which we traditionally thought were fully established by term equivalent age, we can see are still maturing across the term period. On the right here is work from Lucas Franca, showing doing dynamic connectivity analysis, and then showing that there are different brain modes that can be identified with our data, which is really enabled by the high temporal resolution. And then what we're able to see is the transitions between these different brain states, how they're different in preterm infants. And then also, interestingly, Lucas was able to show that there was an association with preterm birth. And so the dwell time, sorry, my slides just keep going. So the dwell time and the kind of different kinds of synchronization and occupancy in these different modes is significantly related to developmental outcomes. So moving on to the fetal data, we acquired 277 fetal data sets from 255 individuals, and those scans were done between 20 and 38 weeks gestation. Again, all this data is publicly available for download. 248 cases passed quality controlled assessment after automated processing pipelines specifically for the fMRI. And we acquired the data on the exact same 3T Philips system, but this time with a 32-channel cardiac coil. The final acquisition has a TR of 2,200 milliseconds with a longer echo time of 60 milliseconds. And we acquire just over 9 minutes of data with 250 volumes with a similar resolution of 2.2 millimeters isotropic. And for that, we use a multiband factor of 3 with some in-plane acceleration as well, sense 1.4. The automated processing pipeline, which again is on GitHub, and Slava Karolis has published a beautiful paper describing it in Imaging Neuroscience earlier this year. It incorporates then the sensory construction. Importantly, it also includes dynamic distortion correction, where we're not just correcting for the spatial distortions just at a single time point, but also considering that there's going to be some distortion in the temporal domain, where if we see ongoing or unexplained changes in distortion, then that's likely to be related to the infant moving within the maternal space, and then the ongoing interaction then with the surrounding tissues. And so then we can unwrap the phase to be able to do a more comprehensive distortion correction, both in the temporal and the spatial domain. We also do slice to volume reconstruction, temporal filtering, including for specific sources of structured noise, which are not just the kind of physiological ones that we mentioned before, but also things like interactions with maternal pulsation and maternal breathing and so on. There's registration into age-specific and group spaces, which you can see on the right here. And again, all these transformers are available for download. And then the data has been packaged into BIDS format for the public release. Slava has then done some beautiful work to try and look at what he can actually pull out this fetal data. On the left here is what he describes as maturational networks, which is a way of looking at networks inside the brain where we can see that the functional connectivity patterns are clustered together. And so they're not resting state networks in the traditional sense. What these are networks of where we can see that connectivity patterns are maturing in the same way. And what's fascinating is that you can see that we can, and then he's done a further clustering approach on that. But what we can see is that there very clearly is similarity across functional homologs in between the two hemispheres. So we have bilateral maturational networks, but also that they cluster together in very similar ways for things that we traditionally would be able to associate with more mature resting state networks, such as the sensory motor system and those involved in perhaps perception and action in later life. Hüppi Willers Moore also looked at gradients within different networks and showed that in the fetal data that we were able to recapitulate the kind of functional networks that you might see in older patients, but also the kind of gradient behavior that you see within those networks. And interestingly, what Hüppi also showed is that you could see very robust gradients within these networks in some of the primary motor and primary sensory networks, but not in the association networks. So I'm just going to move on now to talk a little bit more about understanding BOLD itself. I know I've talked far too long about the DHCP, Petra, so I'll try and really wrap this up very quickly. But the big question is, of course, is there neurovascular coupling in the immature brain at all? Because we're making that assumption when we do fMRI studies. And there has been some particularly important work from Elizabeth Hillman and her group showing in mice that there may not be functional hyperemia at all in the developing brain. And so what they showed is that if you look at adults in their studies, that there very clearly was a rise in oxygenated hemoglobin, which is what we'd expect for a positive BOLD response associated with neural activity. But actually, when they looked at younger mice, that there was not a rise. So the red curve is not going up at this stage in oxygenated hemoglobin, but instead there was an increase in deoxygenated hemoglobin, which really is what we generally consider to be a negative BOLD response. And so their suggestion is that perhaps neurovascular coupling is not mature in the immature brain, and that we should be not using fMRI as an approach to be able to study brain activity. Of course, we're here today to say that we can use fMRI, so hopefully we're preaching to the converted here. But I think the two ways that we try to explore this and really understand this is by looking using simultaneous EEG fMRI. So just to talk about this very, very briefly, what we can see in the developing brain is bursts of neural activity on the EEG, which you can also see in both in mice, but also in preterm infants. And so we've used simultaneous EEG to recognize this bursting activity inside the brain. We can see that they occur across different regions of the brain when we do EEG. And if we use those as regressors for a traditional GLM using simultaneous fMRI acquisition, we can see that we can also then identify the associated BOLD activity with those bursts. And what we found is that we looked at the most common bursts of activity which occur in the preterm brain at around 35, 36 weeks, which are located in the kind of inferior parietal kind of regions, that they were associated with BOLD activity in the intellect that you can see here. We've also looked at the neurophysical correlates of a task. So in this case, again, it was movement of the right hand. So you can see in black here, this is the GFP, that we can see that there's a big rise in the GFP during the time the stimulus is on. The bold response is then lagged and follows that. And then we could also explore the different kinds of microstates that are then occurring or kind of the dictionary of microstates that occur across the period of activation. Lastly, I'm just going to talk about ultra high field fMRI. I can see I'm probably overrunning. But what more can we learn at ultra high field? So with ultra high field, the really key thing is that we can really increase our sensitivity and specificity, and we can really start to explore what happens across cortical depths and within the different kinds of microcircuits that are occurring there. This is really fascinating in infants because, of course, we know that the physiology is changing and we know that the microcircuitry is being established. We've scanned a total of 110 infants now on our 7 Tesla system at St Thomas's Hospital, London. The youngest was 33 weeks post-menstrual age, so 7 weeks before the time of normal birth, weighing just 1.5 kilograms. And this is really work that we've done through development with Shahan Malik and Pip Bridgen, who I've got pictured here. These are the kind of structural images that we get with T2. And just to really just touch on the kind of data that we can get with ultra high field now in our infants. So on the left, this is the DHCP data, which has a resolution of 9.9 millimeters cubed. We are now acquiring fMRI data actually at lower resolution than here. We're now down to 0.8 millimeters isotropic, which really gives us, you can see the kind of detail that you get inside the brain, but also the spatial specificity that you can get with the functional patterns. And this is a work really driven by Yucha Willis-Moore and through enthusiastic collaboration with John Polomeni, who's been a great partner in this work. So we've been using a suite of robotic devices to induce our functional responses, and just to show that these are the kind of functional responses at 7 Tesla compared to those at 3 Tesla. We've been having very enthusiastic support from the Laminaut community and Renzo Huber, who I think is on the call today. And we've been able to then look at the kind of responses that occur across cortical depths. And so we can see that there are indeed developmental changes in the bold response, which are different across depth. And really, we can start to understand how that relates to developmental physiology as well, and changes in vascular density. So I think this is about biophysical modeling, spin echo, and visual responses, but I think I better cut myself off there.
Yeah, we're butting up against time here, but there were a couple questions maybe if people can hang in for just another minute, we can ask. We want to go ahead, Petra, and
yeah, there's actually a question or quite a few questions from Jessica Dubois. First, she thanks the organizers for organizing this great session and the nice talks. But then she goes into questions. Does the neurovascular coupling show differential maturation across functional systems and brain regions? To which extent would this impact the identification of the functional connections with resting state fMRI and the interpretation of their evolution with age? Any comment on the link between the developing functional connections and structural white matter connections, so resting state versus diffusion, and the potential mismatch in the progressive development of short range versus long range connections? So, Tom and Chris.
So the short answer to are there differences in functional systems in terms of neurovascular coupling? I think the answer is yes, of course. I think it also links to a certain extent to Cesar's question about the optimal TE, because obviously that's going to vary across the cortex as well. Because you can imagine the cortex is maturing at different rates. So the kind of vascular density, the kind of water content, the amount of neuronal density is also different across the cortex. And so absolutely, we've got differences in sensitivity across the brain. There are different approaches that we can really take to that. So of course, you can think about using multi-echo approaches, which I think, Damien, you've been doing in Minnesota. You're using multi-echo. We can accept that there's those limitations, and we can try and think about how we then might optimize different acquisitions if we want to answer particular questions. And so we've been doing some work with the visual system at the moment. We've been trying to understand the optimal echo time to use for acquiring data from the visual system, for example, as opposed to traditionally a lot of my work is focused on the motor system, which is much more mature actually than the rest of the cortex. If you look at the echo time, the optimal echo time to use there based on T2 star, we can see that T2 star is much shorter in the motor system as opposed to the, for example, the visual cortex or the frontal cortex. But yes, absolutely. Neurovascular coupling does change. I don't know if Chris and Kevin want to chime in because I'm talking too much.
No, you're muted,
No,
Chris.
Chris, you're muted, Chris.
Sorry. No, you hit the nail on the head, Tom. Just in the interest of time, I think that's nicely encapsulates some of the questions. The structure function question I think that Jessica also raised is germane, and that's where a lot of these data sets, both the DHCP and then the HBCD one that Damien I think is throwing information in the chat, really allows us to uniquely explore. So you can take common data approaches, have the data in a comparable format, and then you can apply different techniques to really lean into that. I think there are some unique insights that we still need to explore in terms of those interrelationships across modalities. But the capacity and methods to do that are now available and shareable. So excited to see where these data sets take us for sure.
Yeah, Kevin, it would be interesting to know because we all have this lateralization in the functional response, but when you look at diffusion, the thalamocortical development of the diffusion tracts is quite symmetric. So where do these differences come? So that's, I guess, related to Jessica's question.
Just to say, Cesar, you were asking about changing the optimal TE across development as well generally. So we thought about this very carefully and we chose not to. We chose to use the same echo time for all of the data. There were obviously limitations to that, but in the end, what we chose to do was try and find a sweet spot, which was prolonged, but was perhaps somewhere in between, but also could balance out the requirement, which we really had, was to try and get a very short TR. So it's kind of balancing all of those different things about your acquisition.
Yeah, thank you. Thank you so much.
So do we go on discussing? I don't see any other questions in the chat.
So I have a comment regarding the motion correction, and I was actually wondering about FIRM in real-time monitoring of motion. So I of course, I'm pretty sure that is using WasU, and Damien is also using it. But what about the rest? Is like, do you have any kind of real-time motion correction? Even looking at the images inline during the reconstruction, I know that with multiband, the reconstruction is a little bit slow depending on the GPU, but what are your thoughts about that?
What I can say is that there's FIRM is good for monitoring, optimizing the scan times, and things for infants. There is we are this close, I think, to, if it's not already available, that develop the actual real-time motion correction for infant scanning. It's part of the HBCD study. I'm throwing a bunch of information about that in the chat as we speak. I don't know if Chris wants to say more about that, but that should be available for people for that particular topic soon.
Yeah.
And I was also wondering whether that's even possible to do it in fetal. fMRI.
Yes,
That would be
I think it would be. Sorry, go ahead.
It's very difficult, but I will say the same people building the version for real-time motion correction during neonatal period and up are working on that solution for the fetal period too. But that is probably a little further away.
So Chris, you want to say something?
Just to maybe potentially what Damien said, the navigator-based approaches for corrections of real-time collections are something that we're keen to roll out. We understand the importance and negative impact of motion across modalities. So there's a effort underfoot to obviously implement that and make it scalable and available. So we're close to having that ready to roll and excited about that because it will pay it forward in lots of different ways, we think.
One question also arises, how are we in the neonatal field to actually have sequences that we can use cross hardware in sort of the open sequence approach? And it seems to me that we're still not quite there yet because many things are system specific developed, especially in the automatic motion correction and so on. So what's your view on that?
Yeah, so we spent a lot of time working on that issue for HBCD because we have sites with scanners across the three major vendors, Siemens, Philips, and GE. Yes, there are proprietary elements of how each vendor approaches that, but we've been fortunate that they've been close collaborators in terms of some of these efforts. We're not going to, I think, in a general sense, get people to provide the keys to the castle in an unfettered way, but I do think that there are ways that we can get common sequences aligned and available that each vendor can support to facilitate this kind of team or shared science approach. So I think there's an intention to do that. I think there's a mechanism for that. And I think we're getting to the point where that's something that's very realistic. And we've tackled some of that, a piece of that pie, but there's still more work to do. And that's where I love sessions like this that are very communicative and collaborative. And, I think we just need to continue to take advantage of these opportunities to get everybody on the same page.
Yeah, and I think ISMRM as a community can be a leader in that.
For sure.
Yeah, and I think some of these efforts and collectives like this can, in essence, help push the vendors into helping harmonize these things. And that's kind of how this got going with HBCD, and it's one design. So the sequences are set up for longitudinal design across 10 years, right? So that's not the perfect acquisition design for all types of questions and whatnot. So there's still a lot more work to do. That's one harmonized sequence across vendors that's available for people, but there's still more work to do for different styles of studies and questions in that front. But collectives like this is how it has to be done.
Perfect. Cesar, you take over or we?
Well, I mean, if there are no more questions,
there is one.
Damien,
star controlling, but yeah. Any comment on star controlling for infants? I guess it's for Tom for the 7T.
Yeah, sure. So for the SAR, we've really and also the guys in Minnesota. So we've had a lot of conversations with Isa Yakub as well in Minnesota about this. We've really taken a very, very conservative approach to it. So we spent a lot of time doing modeling and trying to understand the differences. At 7T, it's actually more of a concern than it is at 3T in terms of the fact that the amount of SAR deposits is higher in infants than it is in adults, which is really the reverse of what we see in adults. Of course, the kind of things that we do with fMRI, to be perfectly honest, are not high sequences anyway. So we're just using gradient echo, 2D gradient echo at the moment. We have used a bit 3D API as well, but we're mostly just using normal API. And so we don't have massive issues with SAR associated with those things. I think that the more concern is when we've tried to do things with spectroscopy and when we've tried to look at spin echo sequences and things like that. So I think from the fMRI perspective with SAR, I think it's not so much of an issue. I don't know, Chris and Kevin, if you guys have really thought of it in the same way.
Yeah, similar. You and I have discussed this before. Yeah, I think it's going to be a 7T issue, but I think in terms of what we do on 3T, it's not really a consistent concern.
Yeah. We have at Minnesota for the infants on 7T, there's you have a process to essentially, model and measure the amount of SAR before any infant goes in the scanner to help be sure that you're not overdoing it with SAR levels, but.
Good to know that you already have experience with 110, so.
It's feasible. So a lot of challenges. So that brings me to the initial point that Patricia did. So a member-initiated symposium on infant and early age MRI and functional MRI could be very, very attractive if there are challenges for the vendors for harmonization of all the techniques and procedures across scanners. Because I guess it's like considering the challenges in the data, the reproducibility, the reliability of the results. I think as a community, we need to work more on these collaborations. Yeah, and also like I would like to also the prospect of multimodal studies. So Tom has addressed about neurovascular coupling, EEG, fMRI, which might be extremely challenging, and I can imagine. I'm also thinking about combining fNIRS with fMRI to know a little bit more, which Chris also has experience in WashU and other teams, of course. But so if there are no further questions, I would like to thank all the panel. I mean, world-leading experts on the field of infant brain function with functional MRI. So thank you very much for accepting to be here. I know that sometimes it's difficult. People are on holidays, so you made the effort. So thank you very much. Patricia, you want to say something?
Not really, just add my thank you to everyone. It was a really great session. Thank you for making it.
Yeah, and we had 46 participants.
No, we had more. We had 72.
More?
They left now.
Yeah. We achieved almost 80 participants.
Yeah, very good.
But
Yeah,
we
very
received
good.
also a lot of emails to say, okay, we can record the session, and then they are on holidays with the families, so they want to join, but sometimes it's difficult. And also, like the schedule that might not be the best time for people in Asia and Australia, or even in the West Coast. So I'm pretty sure that they will. So we will post the recording as soon as we edit it. So we cut the beginning and things like that, and we will post it and let everyone know. Okay.
Great
so thank you very much again.
ISMRM.
Yeah,
Yes.
and see you in Cape Town.
See you in Cape Town, exactly.
Bye, thank you.
Bye.
Thank you
Bye,
all.
everybody. Thanks very much.
Bye.
Bye.
Thank you. Bye.